chloroplasts like mitochondria. They're about the same size as mitochondria, maybe slightly bigger. They're also double membrane, although the orientation of those membranes is slightly different. The inner membrane of the chloroplast is called the thylakoid. What happens here in the chloroplast? Photosynthesis. It turns out that many of the characteristics of the mitochondrion is shared by the chloroplast. It has its own DNA. That DNA is circular. It's gene-rich. And many of the proteins that are required for the function of the chloroplast are actually encoded for in the chloroplast DNA. Furthermore, when you look at the sequence of nucleotides in that chloroplast, it looks very similar, at least for some proteins, to the DNA sequence in photosynthetic bacteria that are still around today. And so, Margulis's explanation for the origin of the mitochondrion is endosymbiosis, and her explanation for the origin of the chloroplast is endosymbiosis as well. What is photosynthesis? It's production of glucose.
Conversion of light energy to chemical energy.
Yes, conversion of light energy to chemical energy. And it turns out that cells make glucose, but in a multi-step pathway. We can break the reactions of the chloroplast, the reactions of photosynthesis, up into two distinct classes of reactions. The first class is called the light reaction. And this is called the light reaction because it is directly involved in harvesting photons from the sun and to converting that kinetic energy in the form of light into potential energy in the form of complicated biological molecules. Again, the series of steps is complicated, but in essence, here's what happens. There's a pigment on the thylakoid membrane right here. That pigment is a pigment called chlorophyll. It absorbs light. It absorbs photons specifically in the blue wavelength and the red wavelength. It reflects everything else, and that's why we see green. Once those photons have been absorbed by the chlorophyll is what we call excited. An electron moves up from one energy level to the next, and when it moves to that next energy level, it can easily be donated to other things. That electron is donated to an electron transport chain, just like we described in the mitochondrion. And it turns out that electron transport chain in the chloroplast has the same basic function as the electron transport chain in the mitochondrion. In the mitochondrion, the electron transport chain is responsible for converting energy in the form of NADH and FADH2 into ATP. In the chloroplast, the electron transport chain is responsible for converting energy in the form of light to energy in the form of ATP. So, in the light reactions of photosynthesis, cells make ATP from photons. What do they use most of this ATP to do? The same things that you use ATP to do. To copy DNA, to power transcription and translation, to maintain ion gradients, to do all the things that cells have to do to be alive. The problem with this pathway is that it relies on light from the sun, right? And so, the light reactions are incapable of making ATP in the dark. What that means then is that photosynthesis doesn't work at night or on remarkably cloudy days? Do cells still need ATP at night and on remarkably cloudy days? They absolutely do. And so, photosynthetic tissues, plants, have the ability to take some of the ATP that have been produced in the light reactions and in a series of reactions called the dark reactions, not because they occur only in the dark, they occur all the time. They're called dark reactions here just because they don't directly depend on light. These dark reactions are also called the Calvin-Benson cycle. It's here in these dark reactions that cells take some of the ATP, not all, it turns out a minority, of the ATP that's been produced in the light reactions and use it to make sugars. So again, in the light reactions that occur here on the thylakoid membrane, cells harness light to make ATP. The dark reactions, which occur here in the stroma or the intramembranous space, cells use the ATP that's been made by the light reactions to make sugars. What do you think they do with those sugars?
Store them and then burn
Store
them
them.
on remarkably cloudy night.
They store them and they burn them when they're needed. How do you think they burn them? In their mitochondria. So, plant cells have two ways that they can make ATP. They can make it directly from the light from the sun, or they can make it from the sugars that they've created as a result of photosynthesis. They can make ATP in the chloroplast. They can make ATP in the mitochondria. Questions about this?
How, I'm seeing ATP to sugar there in the Calvin-Benson cycle, and then we talked about glycolysis, which was basically sugar to ATP.
Sure.
Are they like mirror images?
Well, so if you draw the chemical equations, they're pretty much mirror images of them. Pretty much. If you look at the mechanisms, those mechanisms are not necessarily mirror images of one another. That said, this makes sense in context of the energy diagrams that you draw for 7th graders, right? They make sense in the fact that you draw some picture where there's a sun, and that sun has some funny looking rays that shine on a plant, and that plant makes sugar, where you get your ATP from the plant, right? Or from some animal that ate the plant. And so your ability to get ATP ultimately relies on photosynthesis by plants. So yes, some way, some organism has to be able to fix energy from the environment. We can't do it. Plants can do it. Some other kinds of organisms can do it. But ultimately, all animal life relies on that process. Other questions? It turns out there are various forms of chemosynthesis. There aren't eukaryotic cells, at least according to my understanding, that do this. And so the things that do chemosynthesis are bacteria, and bacteria don't have organelles. Bacteria can do photosynthesis though, right? And they can do cellular respiration. Where do you think they do it? I talked about membranes as being really important because they limit diffusion, right? They keep bad things out, they keep good things in. They concentrate the molecules that are necessary for life. right? Think of this room as a cell, and all of the walls and the ceiling and the floor as being the membranes. These membranes keep us in, right? In addition to limiting diffusion in 3 dimensions, membranes also do a really, really good job of limiting diffusion in 2 dimensions. So now imagine a scenario in which you were in the room, but you were physically linked to the wall. And so that you could move around the wall and the ceiling and the floor, but I couldn't move from here over to that window. It turns out the molecules that are involved in photosynthesis and cellular respiration, the electron transport chains of photosynthesis and cellular respiration, are performed by proteins that physically interact with the membrane. What is the effect of limiting diffusion in 2 dimensions versus 3 dimensions? Imagine 2 ping pong balls in this room, right? And they're bouncing around randomly like the old screensaver used to be, right? What's the likelihood that they come together relative to the likelihood that 2 ping pong balls that are in the wall and moving in the wall come together? It turns out the likelihood that these ping pong balls diffusing in a wall would randomly collide is significantly higher than the chance that ping pong balls in the room would bounce together. And so, if you build machines in membranes, you increase the likelihood that these molecules will actually come together and perform some task cooperatively. And as you might suspect, complex biochemical pathways like photosynthesis and cellular respiration are complicated processes that require multiple players. And so it turns out that things like photosynthesis and chloroplasts occur in membranes for that particular reason. And so, in synthetic organisms, they have similar kinds of pathways in that they're able to fix energy from the environment, although mechanisms are different. But it is routinely the case that those energy fixing mechanisms are limited to membranes. And in bacteria, where are the membranes? There's one, right? It's the plasma membrane. So those chemosynthetic organisms don't have organelles, but they have a membrane in which these machines can be built. And it turns out the machines are generally built in membranes for the same reasons that the photosynthetic and cellular respiration machines are built there. Does that make sense?
Yeah.
I took a very simple question with a pretty simple answer and talked far too long about it, right? And so it's not that complicated. It occurs in the membrane. And it occurs in the membrane for the reason that I just described.
So in bacteria, they do all of these processes. They can set up like an electron gradient across their cellular membrane.
That's exactly it. That's exactly it. Are there differences between the proton motive force, the electron gradient that's produced in photosynthesis in chloroplast and photosynthesis in bacteria? There are, but they're small. Are there differences in the proton motive force that's set up in bacterial membranes and in mitochondrial membranes? There is, but it's small. They do the same exact thing with some slight differences. I would argue that's another statement in support of endosymbiotic theory, right? Where do you find energy production? Well, you don't, it's not only that you find it in the mitochondria. You find it in the inner membrane of the mitochondria, which according to endosymbiotic theory corresponds to the plasma membrane of the internalized cell. Does that make sense? I like the proton motive force a lot, right? And so, just a quick take home. I don't want to go through this in detail because it'll take a long time. But if you can use ATP to drive an ion gradient, you could take a Thronev ion gradient to drive ATP synthesis, right? In theory.
Mm-hmm.
That's how the mitochondria works. Really, everything that's done to convert energy in the form of NADH and FADH2 is invested in making an ion gradient. You make this really, really, really steep ion gradient, and then you use the energy stored in that energy position to make ATP. Turns out you do that not only in the mitochondrion to make ATP, but you do it in the chloroplast to make ATP as well.